Welcome back to the Classics of Immunology Journal Club. Before we get started, don't forget to like the video on the YouTube site and click on the bell to get notified of others. Subscribe. Check out my website. And on the writing tab on the website, you'll find all the classic papers, including the one we're going to do today. The one we're going to do today came out in PNAS, the Proceedings of the National Academy of Sciences, in February of 2006. The authors are Deborah Stauber, Eric Debler, Patricia Horton, myself, and Ian Wilson. And the title of this paper is The Crystal Structure of the Interleukin-2 Signaling Complex, a paradigm for a heterotrimeric cytokine receptor. Now, the background on this paper is that Deborah Stauber and Eric Debler were the first and second author of the paper, and they contributed equally to the work that went into this paper, which went in for quite some time, actually. They were both trainees with Ian Wilson at the Scripps Research Foundation out in La Jolla. And Ian is the senior author, and he's the critical structural biologist. And his background is that he's from Scotland, and he went to the University of Edinburgh, where he graduated with a bachelor's in biochemistry. And then he went on to get a PhD in molecular biophysics from Oxford between 1977 and 82. So that was 5 years at Oxford. Then he did a postdoc with Don Wiley at Harvard. And that postdoc was very productive and really basically made Ian Wilson's career because he solved the structure of the hemagglutination protein of influenza virus, which was a big deal at that time, still is. So the background on this is that we had first identified the IL-2 receptor through classic hormone binding assays using radioisotope labeled T cell growth factor at the time, before it really became an interleukin-2 molecule. But we purified that to homogeneity, had internally radiolabeled it with radiolabeled amino acids, purified it, and then developed a binding assay and first reported that in 1981. And we started then thereafter trying to figure out how to go to the next step and find out how this receptor was working and what was going on and so forth. And as you probably know, there was an ensuing saga that took place in terms of trying to understand the molecular structure of the receptor. And it turns out it's comprised of three different chains. The first was identified only 1 year later in 1982 by ourselves in collaboration with Tom Waldman's group, who had developed a monoclonal antibody that was called TAC, or anti-TAC, that ultimately identified the first chain of the IL-2 receptor. It wasn't until then, in 1987, 5 years later, that the second chain, the beta chain, was found by ourselves, Waldman's group, as well as Warren Leonard's group. So three different groups came out with almost simultaneous papers, 1 and 2 months, 3 months apart, the end of 86, the early 87. And then it wasn't until 92 when Kazuo Sugimura identified the gamma chain of the IL-2 receptor. And then soon thereafter, the gamma chain was found to be mutated in patients with X-linked severe combined immunodeficiency. And of course, that then led to the development, really, of a whole new field of inborn errors of immunology, just like we used to have inborn errors of metabolism when I was a student and growing up. And now there are so many mutations, particularly in the cytokines, their receptors, the biochemical signal transduction pathways, and so forth, that are responsible for many genetic mutations that lead to primary immunodeficiencies. The other thing that came out of all this work was the fact that the gamma chain, when it was after it was cloned and sequenced and so forth, was found to be involved in several other cytokine receptors. As a matter of fact, 6 total cytokines receptors employ the gamma chain as one of the signaling components. So the question was, how can that arose out of this that we'll touch on today is, well, how can one receptor chain be involved in so many separate and distinct cytokines? Because even though they were homologous in terms of their amino acid sequences, they weren't identical. And so that was sort of a conundrum that was really necessary to be figured out and solved. Now, in the introduction of this paper, they go through a very nice summary of where the state of the art as in 2006. And of course, one of the things I wanted to mention was that so we started collaborating with Ian Wilson back in 1985, 86 to try to get to the structure of the IL-2 receptor. And of course, this is before we realized that there was a second chain and we had data for a second chain. We invited Ian to come out to Dartmouth, and we had a conference center that was called the Menary Conference Center. It was on Squam Lake in the middle of New Hampshire, in the foothills of the White Mountains. And actually, that's the place where they filmed on Golden Pond with Jane Fonda and her father. Is it Peter Fonda, I think? Anyway, we wanted to explain all the work that we had done to Ian Wilson, and we were hoping to seduce him to collaborate with us so that we would be able to approach this structure. So, so it took 20 years for everything to be sorted out and to be figured out and so forth. So we started in 85, 86, and it wasn't until 2005, 2006 that we finally got what we're going to talk about today. One of the critical experiments that they talk about in the introduction, or we talk about in the introduction of this paper, were experiments done by one of my graduate students, Hui-Mei Wang, who was originally from Taiwan, but she was a Dartmouth biochemistry graduate student. And she did very critical and very nice studies of both the equilibrium binding to the three separate chains of the IL-2 receptor. Actually, only two of the chains at that time. This is now 86, 87. And she showed when she did kinetic binding studies, and that is determined the on rate and the off rates of interleukin-2 interacting with the receptor chains, that the on rate to the alpha chain was so fast it was hard to measure. And that was combined with an off rate that was just about equally as fast. So it was going on and off very, very, very rapidly. And that was sort of a strange thing because it was, in the end, the equilibrium dissociation constant of IL-2 and the alpha chain was the lowest of all in the end, that we figured out. And what Hui-Mei found, we called her Amy too, I guess that was her anglicized name, is that the beta chain, by comparison to the alpha chain, the on rate was much slower and the off rate was much slower. But then when you combined the two, or had both of them expressed by the same cell, you had a very fast on rate and a very slow off rate. And that makes for high affinity, because what happens is that the receptor grabs the ligand and doesn't let go. That's high affinity. And so one of the things that this paper that we're going to look at today deals with is how that happens from a structural standpoint, the so-called structure-activity relationships. I think we can go to screen sharing right away, and I'll show you some of the results. Yeah, so this is the paper that came out in February of 2006. This is figure 1, and this is a ribbon diagram of interleukin-2 bound to the three chains of the IL-2 receptor. And so just to give you some orientation, in A, this is a side view, looking at the interleukin-2 receptor complex from the side. And in B is a top-down view. And in yellow is the IL-2 molecule and the four anti-parallel alpha chains of the IL-2 molecule. Here in magenta, we have the alpha chain coming down from the top. And over here, when you see the top-down configuration, here is the alpha chain. And over here, then, is the beta chain with two different domains. Each of the chains had two different domains, domain 1 and domain 2. And you can see that IL-2 is being bound by what they call the elbow region between the domain 1 and domain 2 of the beta chain. And it's a similar situation with the gamma chain here, with domain 1 and domain 2, and the interface between IL-2 and the gamma chain was the smallest area of any of the interactions that we found. Another interesting thing about the beta gamma interaction between those two different chains was that there was a huge, very large area of contact between the domain 2 of the beta chain and domain 2 of the gamma chain. There was another group that actually beat us into publication that came out in Science in 2005, in November of 2005, and this happened to be Chris Garcia, who had been a student with Ian Wilson at the time that Ian had started these experiments way back in the 80s, early 90s. Similar configuration of a ribbon diagram here. Here now, IL-2 receptor alpha is on top here. IL-2 is in magenta, and we have domain 1 and 2 of beta and domain 1 and 2 of gamma. And you can see that the interface between beta and gamma is quite striking. I like this particular representation here because now you see that alpha is interacting with interleukin 2, but it's not really interacting with beta or gamma, and you can see that here as well. So IL-2 receptor alpha doesn't interact with the other two chains of the IL-2 receptor. Let me just see if I've covered the whole thing. Yeah, I think that does it. Oh, wait, I want to show you one more thing and another view. Now this is a space filling model of the structures. And since now this is a side view, we've got alpha up here, and we've got IL-2 in magenta, we've got IL-2 beta here, receptor beta, and receptor gamma here. And so that really emphasizes this close interaction between the beta chain and the gamma chain, and the fact that alpha doesn't really come into close contact with either the beta chain or the alpha chain. That's it from the standpoint of this data and the screen share. You should read both of these papers, because there's a lot more data in the papers that are difficult to represent in the discussion of this paper. They go into in detail that because the alpha chain does not interact with the beta or the gamma chain, it seems, and because it's a very fast on and off rate with the alpha chain and IL-2, what alpha is really doing is serving as a ligand capture and carrier to the rapid on rate, facilitates together with the large excess of alpha chains on the cell surface. That was another thing that we discovered and reported on back in the mid 80s. There was always a 10 to 20 to 100-fold excess of alpha chains on the cell surface as compared to the beta and the gamma chains. And so what's going on is that this alpha chain is serving as a capture mechanism and bringing the ligand into essentially into the two-dimensional plane of the membrane, and where it initiates then interaction with beta and gamma. And so based on what the structures and the interaction between the different amino acids of the ligand and the receptor chains, it was surmised that what's probably going on is that the after the capture of IL-2 by alpha and then the bringing in together in a sequential manner the beta chain, the interaction between between IL-2 bound to alpha and beta facilitates a configurational change in the contact region between the beta chain and the gamma chain. And that then brings in the gamma chain into the quaternary interleukin 2 receptor complex and initiates signaling, because the gamma chain and the beta chain then both get activated. And so that's basically what they figured is that this serves as a safety mechanism against premature signaling via an IL-2 alpha beta dimer or a beta gamma dimer without IL-2. That wouldn't be good, since it has to be a hormonal initiation of this whole process. The other thing that they go into in the discussion is how the interleukin-2 gamma chain receptor gamma chain, how that can be involved in 5 other cytokine receptor situations. And they point out in their data from the IL-2 receptor and binding and so forth, is that the interface between IL-2 and the gamma chain is a very small interface, and there's only a few different inner bonds between the amino acids that might be involved in this whole thing. And they think that the specificity of the gamma chain with the beta chain and the large area of contact is really what's important for the gamma chain and how it can then be a promiscuous chain, if you will, and function with these 5 other cytokines. So that's the story with regard to the structural biology from 2006, almost 20 years ago now. There's subsequent things that have happened in this business that relate to this. Is in 2010, I collaborated with Gregoire Altan-Bonnet. It's at the Sloan Kettering. I was at Cornell in New York City, and owner Fienerman, and they did a series of experiments looking at regulatory T cells, because regulatory T cells have a very high density of the alpha chain on the cell surface. And what they found and what they showed is that the marked increase of the alpha chain density on the cell surface increases the affinity of the whole IL-2 receptor interaction markedly, to the point that the equilibrium dissociation constant goes from 10 picomolar to like 10 femtomolar, 10 to the minus 15th molar. Well, that gives the Treg cell distinct advantage, particularly when there isn't very much IL-2 around. The Treg cell can bind, internalize, and degrade IL-2, thereby thereby being a sink for IL-2 and taking it away from effector T cells that are trying to have a meaningful immune response. So that's really some of the most important things that have come out of this whole structural biology emphasis on this business. The other thing that's happened just recently, within the last year or so, is, and actually I should back up and say, in addition to Ian Wilson that we collaborated with back in the mid-'80s, I collaborated with Tom Ciardelli, who was a protein peptide chemist. And he immediately spent a decade or more looking at the sequence of interleukin-2, trying to make interleukin-2 congeners, hoping to be able to get antagonists of IL-2, and also even perhaps better Darwin, and get better agonists than IL-2 itself, so that they could be used therapeutically. At the time, we collaborated with 2 other people that were involved in protein structure prediction: Fred Cohen at the UCSF in San Francisco, and Howard Howley, who was a former IBM computer specialist. And Howard and Fred and Tom all were working on using artificial intelligence, neural networks, to try to take because this is one of the fundamental things about life. The DNA sequence encodes the amino acid sequence. The code of the amino acid sequence dictates how that long chain of the protein is going to fold up into first a 2-dimensional, which Tom and Fred solved, a 2-dimensional structure and then into a three-dimensional structure, because it's a three-dimensional structure that actually binds to the receptor and has the biological activity and so forth and so on. Well, you know, as I've said, as I used to say to my students and postdocs and anybody who would listen to me, anything can happen in science. Anything's possible, but some things take a long time. So that was the mid '80s at Dartmouth, and now the computer artificial intelligence people have cracked the whole field of structural biology wide open, because they have methods now with big data and so forth, which has been called deep mind, and it's the using neural networks and artificial intelligence to be able to do what Tom and Fred and Howard were trying to do, and myself trying to do back in the '80s. And so keep your eye out for deep mind, because it's going to make the structural prediction of just about any protein you want to imagine within the realm of possibility. One of the reasons that it took so long for Ian to, in addition to the fact that we didn't know that we were dealing with 3 different chains of the receptor, to crystallize all 4 of these molecules together, and then to get really good crystals so that you could get the data that you needed in order to be able to calculate where all the atoms were in these molecules. So I'll stop there, and thanks for coming. Don't forget to like the video and subscribe, click on the bell, check out the website, and come back next week. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.